Hello everyone, welcome to the new session of hybridization in plant breeding. That is the second part. We have already covered the hybridization in plant breeding part 1. Okay, so coming back to the procedure of hybridization. So there are 7 different steps in the procedure of hybridization. The first one is selection of parents, selfing of parents or the artificial self-pollination, emasculation, bagging, tagging, crossing, harvesting and storing the F1 seeds, and raising the F1 generation. So let us see this in detail. The first step, that is the selection of parents. So based on the objective of the breeder, the parent's plant must be selected. So what is our objective? The first thing, our objective, we will select as a breeder, we will select the parent plant. Okay, so it is also assured that the parental plants for the hybridization must be selected from the local areas and are supposed to be the best suited to the existing environmental condition. So why we are taking plant from the local areas? Because this local area is the existing environmental condition. This plant is living in this existing environmental condition, and we want a new plant, a new variety of the plant in the same environment. We have to grow that particular plant in the same environment. So we must select the parent plant from this particular local area only, because that particular hybridized plant or the hybrid plant will be grown in this area only. So that should be matched with this. Okay, that should be able to grow in this environmental condition. So it is suggested that the selected parent must be selected from the local areas and are supposed to be best suited for the existing environmental condition. For example, to transfer a gene for resistance to drought means this particular environmental condition, that is a drought. Less water content is here or less water availability is here. Humidity, salinity or the donor parent is selected. Okay. Next step is the selfing of parent, test cross or the artificial self-pollination. So it is essential for inducing the homozygosity. The parent which we are selecting must be homozygous. Suppose I'm taking this particular plant, capital A, okay, and the other plant is capital B, small b. This is again hybrid one. So my parent means this particular plant, selected parent plant must be homozygous, capital B. So what we must do? We must get this plant. Selfing is done. Okay, selfing of the parent, that is the test cross or the artificial self-pollination. Test cross means this plant will be taken, capital B, small b, and it is pure recessive one, that is a small b. When it is done, then you will get capital B, small b and small b, 2 different types of parent, and also capital B. So in the test cross, recessive parent is taken and this particular plant is selfed with that. So it is essential for inducing the homozygosity or eliminating the undesirable characters or obtaining the plants. Therefore, a test cross should be done after F1 generation to get homozygous generation. Now, the third step, that is the emasculation. The plants are grown under the normal conditions and are emasculated. Okay, so the plant which we obtained after the F1 generation must be grown in the normal condition and emasculated. What is the emasculation? Emasculation is the removal of stamens or anther or the killing of the pollen grains of a bisexual flower without affecting in any way the female reproductive organ. Okay, so we are removing the stamen or anther or killing the pollen grains, but be sure that there should be no pollen grain present in the anther. So hand emasculation. Now there are different methods of emasculation. The first one is the hand emasculation method. So this method is generally used in those plants which have large flowers. So with the help of forceps, we will take out the anther as shown in this figure. Okay, with the help of hand only and forceps. So this is done by opening the corolla of the selected flowers and removal of stamens using the forceps carefully. So that is why it is called as hand emasculation method. Flowers should be selected at the proper stage. Stigma should be receptive and anther should not have daces. All the anther should be removed. No anther should be there. All the anther should be removed from the flowers without breaking it. Stigma and ovary of the flower should not be damaged. Removal of, now the second step or the second type of emasculation, that is the hot water treatment. Removal of stamens with the help of forceps is a very difficult in minute flowers that we can do in a larger big flowers. But what about in the case of minute flower? In small flowers like bajra or jowar, emasculation is done by dipping the flower in hot water for a certain duration, that is 1 to 10 minutes of a time. So this is the done in the hot water treatment. However, the time varies from species to species. Now this time which is given, this is not for all the species. It varies from species to species. This method is based on the fact that gynecium, that is the female reproductive organ, can withstand the hot temperature at which the anthers are killed. Okay, cold water treatment. Now the third type of emasculation. This method is less effective than the hot water treatment. Like hot water, cold water also kills the pollen grains without damaging the gynecium. Gynecium is withstand with this cold water treatment, but at this particular temperature the pollen grains are killed. In rice, 0 to 6 degrees centigrade temperature is maintained to kill the pollen grain. So this method can be adapted for the rice. Alcohol treatment method, fourth type. This method is not commonly used for the emasculation because duration of the treatment is an important factor, since a very short duration is required for failing which even the gynecium may be damaged. Okay, flowers or inflorescence are immersed in alcohol. So we will take the flower or the inflorescence and we will totally immerse that flower in the alcohol of a suitable concentration for a short duration of time. Right? Like in alpha plant, a treatment is done just for 10 seconds and 57% alcohol sufficient to kill the pollen grains. So at that particular concentration, gynecium do not get killed, but the pollen grains are killed in the case of alpha. So this method can be adapted for the alpha. Suction method, fifth type. It is a mechanical method and it is suitable for the crops having minute flowers. Okay, so in this method, amount of pressure is applied in such a way that only anthers are sucked out and other parts of the flower like gynecium remain intact to the flower. So that is why it has got its name, suction method. This method is 10 to 15% self-pollination takes place. In this method, only 10 to 15% self-pollination takes place. Male sterility or self-incompatibility method. Now this is the another type. Emasculation can be eliminated by the use of male sterility plant. Okay. In some self-pollinated plant, for example, sorghum, onion and barley, anthers are sterile. Okay, anthers are completely sterile and do not produce any viable pollen. So there is no need of doing emasculation. Okay, similarly, the self-incompatibility may also be used to avoid emasculation. So this is one of the advantage of this method. Chemical method. Certain chemicals are capable of causing male sterility when sprayed before flowering. Okay, 2,4-D, naphthalene acetic acid NAA, malic hydrazide MA, and tribenzoic acid, etc. These are some of the chemicals that are used for causing male sterility in the plant, making the anther or the pollen grain sterile. FW450 in a cotton may be used in bringing about the emasculation. So this is the chemical method. Now the next step, that is the bagging. After emasculation, there is a process called bagging. The bagging is done with the emasculation in the bisexual plant, and before the stigma is receptivity and the dehiscence of the anther is in unisexual plants. Both male and females are bagged separately to prevent contamination. Now bagging is done just to cover your male and female flower because you want to do the artificial pollination. So the pollination should not be done by its own, right? So that flower or the emasculated flower should be covered with the help of polythene bag or cotton cloth. So in this, that prevent contamination in male flowers and cross pollination in the female flowers. The bag should be made of paper, butter paper, or even sometimes fine cloth is also taken. Butter paper or the vegetable parchment bags are most commonly used in this method. The bags are tied to the base of the inflorescence or the stalk of the flower with the help of a thread. So here we can see, now this is tied here at the base. This is again tied here at the base. And tagging is also done. Now the tagging. The emasculated flowers are tagged just after bagging is done. The tags are attached to the base of the flower or inflorescence with the help of thread. The number referring to the field record, date of emasculation and date of crossing, all is mentioned in that tag. So we must do this tagging, the date of emasculation, the date of crossing. Okay, so all is written on that tag and this is called the tagging. Now comes the crossing. In the sixth step, it can be defined as the artificial cross-pollination between the genetically dissimilar plants. Now we will have one male flower and one female flower which we have bagged and tagged already. Now we will remove that tagging and we will do the crossing, and this is called artificial cross pollination. So in this method, mature, fertile and the viable pollen grains from the male parent are placed on the receptive stigma of the emasculated flower to bring about the fertilization. Okay, so pollen grains are collected in the petri dish. We will collect with the help of brush, okay, or forceps. This pollen grains are collected in the petri dish. For example, what are the plants which we take? Wheat can be taken, cotton, etc. And the paper bags, that is in the case of maize, and applied to the receptive stigma with the help of brush or forceps. Now, the next step is the harvesting and storing the F1 seeds. The seeds of the cross plants are harvested. Okay, we will now, when the seeds are produced, we will take out those seeds, that is called as harvesting, and threshed or even drying. Seeds are stored and tagging should be done properly. We will store those particular seeds, okay, and we should also tag so that we should not get confused with the other stored seeds, right? Next is raising the F1 generation. In the coming season, the stored seeds are sown separately to raise the F1 generation. The plants of the F1 generation are progenies of the cross seeds and therefore the hybrid. So once the seeds are stored, after we will have a suitable season for that particular crop, we will take out those stored seeds and we will raise them separately. Okay, to ensure that these are those hybrid plant that we came after the hybridization process. Okay, so the plants of the F1 generation are the progenies of the cross seeds and therefore we will get the hybrid plant. Okay, so there are the various methods of the improvement of self-fertilized crop by the hybridization: pedigree method, bulk method, single seed descent method, back cross method, and multiple cross method. So these are the different improvement of the self-fertilized crop by the process of hybridization. So this is all about the hybridization. Okay, thank you very much.